Get into viruses, which I know everyone at the time of this recording is interested in because we're still in the middle of the COVID-19 pandemic. So this is going to be chapter 6 in your textbook. In your current textbook that the department's using doesn't go over viroids and prions, but I'm going to throw them into here, so you'll have this piece in lecture. I think maybe your book talks about prions perhaps, but definitely not viroids. And I'm going to throw them into this lecture, even though viroids at the least is not in your textbook, because they are a related topic. So viruses are that gray area between living and non-living. They're not technically alive, but they have some lifelike qualities. Prions and viroids would fall into that category as well. They are infectious particles. They can replicate, but not on their own, and they're a gradient between life and non-life. And I think it's just interesting to see this full gradient, even though some of this isn't going to be in your textbook. And I'm not going to test you on viroids because viroids are exclusively plant pathogens. So I won't test you on viroids, but I still think it's really important to see this full gradient because it gives you a clearer concept of how, or visual, I guess, of how life may have arose. You know, what does it mean to replicate? What are the implications of self-replication, and what are the many ways that this can happen? You know, because we can't see the microbial world, and even of the world that we can see, we still get stuck into the way in the thought process about how things work for humans. You know, we get stuck on two sexes. You know, like most of the time there's male, female. For some people, you know, under some people's beliefs, that's literally all there is, even though there are some other things that can happen. But you know, you get into this two-sex system. From a biological standpoint, and you don't even recognize that there are some animals that don't have two sexes, and there are plenty of fungi that don't have two sexes, as we've already discussed. You know, the whiptail lizards of New Mexico, they're all female. There are 0 males. I always feel like the whiptail lizards should be New Mexico's mascot, because you know, the Virgin Mary is really important in Mexican Catholicism, and these are all female lizards. Right, so they're all virgins, and they also have that quintessential turquoise look that is so, you know, New Mexico is sort of famous for its turquoise jewelry. So I always feel like that should be the mascot of the state. But I'm just trying to make a point that, you know, looking outside of your wheelhouse, looking outside of your bubble of concept can really help you understand how the world is actually put together. Because we walk around with these blinders every day with our only partial truths about how life is put together. And so in this class, the usefulness, I think, of the microbial world from a philosophical standpoint is to break out of your mold and start understanding, you know, get a clearer picture of what life really means by looking at all of these other examples that break the mold that we're used to. So viroids are only plant diseases, and like I said, they won't be on the test. They're just tiny segments of RNA that are internally paired to create this three-dimensional shape. So by internally paired, I've told you that RNA is single-stranded. This is a single strand of RNA, and it's just a tiny, tiny strand of RNA. And it is a single strand, but it pairs up on itself. So it can fold back. Even if you have a string, you know, you can fold it back and tie knots or whatever. So this RNA has a three-dimensional shape, sort of like a protein does, by internal base. And that internally paired bases, that they follow the complementary base pairing rules. So U is the equivalent of T, right? U in RNA is the equivalent of T in DNA. So U to A's and U's always pair up, and G's and C's always pair up. So those complementary base pairing rules, they dictate how this RNA strand is going to fold upon itself into a three-dimensional shape. Now, what a viroid does is it infects a plant cell and it uses the host's RNA polymerase for replication. So remember, RNA polymerase is that's an enzyme that creates RNA. So ACE means enzyme. Polymer is a long molecule made out of repeating subunits, as we talked about in the previous lecture. So RNA polymerase is the enzyme that builds RNA. So what the plant host's RNA polymerase does is it reads the viroid RNA, and it replicates it. So the plant is actually so this is not, this is not it's not a truly self-replicating particle. It's it needs the host cell. It needs the equipment carried by a living thing, by a living cell, in order to replicate. It needs the plant cell's RNA polymerase, because it's just a piece of RNA. It's not carrying anything with it. There's no satchel, right? Like the cell is one giant satchel, bound by the plasma membrane. These viroids are just exposed RNA. They have no protective casing. They don't carry anything with them, so they need the equipment from the cell. But the cool thing about viroids, the thing that really makes them interesting, is that they have some catalytic functions, which means that they are enzymes. So they are two things in one. They are genetic instructions that can be replicated, right? Genetic instructions, and they are also an enzyme. They are two things in one, okay? They are the blueprint and the enzyme. So in living things, the two molecules have different roles. So nucleic acids like RNA and DNA, in living things, they carry genetic codes, right? And then you have proteins that are enzymes almost all the time. But in viroids, they're interesting in terms of this gray area between life and non-life, because they have more independence than any other gray area that we see, because they have, in one molecule, you have the catalytic functions of an enzyme and the blueprint that can be replicated. All right, so scientists are interested in that because it seems a little bit like precellular life, doesn't it? So it's not precellular life itself, right? Because you can't have a viroid before you have a cell, because the viroid relies on the cell to replicate. But scientists are interested in viroids because in the search for what the first life might have arisen out of, like we can see cells in the fossil record, but we can't see the things that came before cells. Some people, of course, believe that God created everything, and that's fine. But for those who are looking for the evolutionary origin of life, even if it's there, we're not going to see it in the fossil record, because the molecules that are smaller than cells, they're too small to fossilize. So we will never find the self-replicating molecules that predated cells, because they are too small to have an imprint in the fossil record. They could not. A single molecule cannot be fossilized. So something like a self-replicating piece of RNA, it's not going to be fossilized. Even if it was there, it's not going to be fossilized. So what we have to do as scientists is sort of think about, like, what are the examples on the planet today that could give us some clues about what might have happened before cellular life. And viroids are one of those interesting examples because of that fact that they have this very a lot of independence in terms of replication in one molecule. So it is not precellular life, but it is an example of how the only thing that would have to be different is it would have to be able to replicate itself instead of relying on the cell for replication. That's the piece that's missing. Right? We don't have any examples of that in the natural world today, but scientists have been able to create self-replicating RNA molecules in a lab, but we don't have examples of them in the world today. Okay? And so viroids are very similar to that. So this is just a picture of how this looks. So you have the viroid genome, and so another thing, it's a small bit of RNA, and I said it's single-stranded, but it's actually a loop, sort of like a bacterial chromosome, except it's RNA instead of DNA. And it folds back on itself, but if you unpaired these bonds, it would just be a circle like you see here. So what this does is it infects a plant cell, and there's the host's RNA polymerase right there in gray. So that's the little host, that's the host cell, the plant cell's enzyme that replicates RNA. And so what it does is so this viroid unfolds so that it's just a loop, and then the host's RNA polymerase is going to read it and create a new strand of RNA, and that's that little tail coming out there is the new viroid being replicated, and it comes off. But now the thing is the reason this is a pathogen is because because this is a loop, there's no end, right? So the host's RNA polymerase is now forever stuck on this loop, just creating more and more and more and more of this viroid. So it goes around, 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 around. And viroid genomes, RNA genomes, just keep getting spat out like a tape, right? Now, these viroid genomes are just in one long line and they're all still connected, right? Because the host RNA polymerase is just going around and around over and over again without stopping, so it creates the new viroids all as one piece, all stuck together. So what the viroid, this is the catalytic, where the catalytic functions of the viroid comes in. So here they are, all these viroids. They cleave themselves. So self-cleavage. Self-cleavage means this is the end. So here's the end of one viroid and the beginning of the next. So it's able to cut itself between viroids. It cuts itself. So here's one viroid, here's number 2 viroid, here's number 3 viroid, and it'll cut itself into single viroids, so it'll separate itself into the separate viroids, and then it'll seal its own ends back together and fold itself up this way. So there's where you see it has these catalytic functions like an enzyme, and it is also a bit of genetic material. And then these new viroids that all fold up, they go on to the next cell or to the next RNA polymerase and get the next one wrapped around. Now this is a virus because, this is a pathogen. It's not a virus, it's a viroid, but it's a pathogen because even though it's not making a toxin that would make the host sick, what it's going to do is it's going to keep making more, this RNA polymerase is making hundreds of these, thousands maybe. It just keeps going around and around and it will never get off the loop. And then all of those thousands go to the next RNA polymerases and they get stuck on the viroid and they can never get off the loop. What that means is if these RNA polymerases never get off the loop, these RNA polymerases are now not functioning for the plant. And what role do they have for the plant? Well, transcription, which is really important for DNA expression. So we just talked about earlier this week how gene expression is taking the genetic instructions in the DNA of the plant or of the animal or of the bacteria, whatever the living thing is. You have the DNA blueprint, which is the instructions for building life, and then gene expression is expressing that blueprint and actually building something from it, building the proteins from it. Now, in that process, gene expression happens in 2 steps, transcription, translation. Now, if you don't get transcription of DNA to RNA, then you don't get translation of RNA to protein, right? So if all of the host's RNA polymerases are stuck on these viroids, then they can't transcribe the plant's own genome. They can't express the plant's own genes. So now the plant stops making proteins. It stops being able to express its DNA. It stops being able to use its own DNA. And now the plant is, the plant cells are going to die because they are going to need to make proteins. They're going to need to have enzymes for their metabolic functions. They're going to need to be able to build cell parts and repair. They're going to need to be able to replicate. And if all and they're going to need proteins to do all those actions. So if their RNA polymerase is wrapped up in these viroids, then their RNA polymerase can't serve the plant. And you could go all the way, so then the cycle repeats.